Good morning, everybody. Let's get busy. We have some of the big stuff in meiosis to cover today. All right, so we finished up, I think about here. All right, so come on, type not working today. Two big things happen in meiosis. Meiosis 1 specifically. Anybody think what they are? At least one of them. What's the... Actually, yeah, there's crossover. I was just thinking, oh, not that you're wrong. I just think that's probably actually 3. That's 1. Also called recombination and synapsis. What else? What else happens in meiosis 1, or by the end of meiosis 1? Yep, diploid to haploid. That's a really, really big thing. Right. And the final thing, I'm not 100% sure if we cover this, so don't worry if we haven't, we'll go over it. But there's another one really big deal that is essentially the basis of the law of independent assortment. Anybody think what that might be? Particularly if you look at the metaphase picture down at the bottom left of this slide. What about those chromosome pairs? Are they lined up in that particular way for a reason? Will they always be lined up that way? Have a think. Good guess. Sorry. It's hard to answer these questions when, we haven't covered it, which I don't think we have. The answer is no. So the other big deal is the random alignment of... I'll be so glad to finish this where I don't have to type out homologous anymore. Chromosome pairs on the metaphase plate of metaphase 1. So the image that you got down there of metaphase 1, that's one possibility. The other possibility is that they're the other way around. And we've got to think like what the daughter cells are getting. The other thing is they might be opposite or the other way around, right? So for each chromosome, chromosome pair really, there's a 50-50 chance it'll be one way around versus the other. Now you might be thinking, well, like, why does that matter? It matters because homologous chromosomes are not identical. They are very, very, very similar, but they don't have exactly the same alleles for each gene. Yeah, absolutely. That is exactly what's going on. And so this is for every single chromosome pair. So if you think like this is, the chromosome, remember these are sister chromatids. So this is one chromosome from your mother, and this is one from your father, right? They could line up on the metaphase plate like that. Or they could line up like that. And so when those get pulled apart, which chromosome of the pair the daughter cells get is random. Could be this daughter cell gets the maternal chromosome and this one gets the paternal. It could be the other way around, right? It just depends on how they get caught by the spindle fibers and pulled to the center. And so I kind of, not being super organized today, but think about it this way. If you have two possibilities for every chromosome, for this cell that has two chromosomes, you have 4 possibilities. Well, 2 pairs of homochrons. So that's 2 to the power of 2. For us, right, for humans, we have 23 pairs of homochrons. Chromes, dang it. You have 2 to the power, there it is, 23 combinations equals 8.4 million. So every single time you have one of your germ cells go through metaphase 1, you have 1 in 8.4 million possible combinations of how all of those chromosomes, for each one of those 23 pairs is either that way around or that way around, that way around, that way around, that way around, that way around. Totally random every time, right? And so this alone, even before talking about crossing over or recombination, this alone generates an enormous amount of diversity in the gametes, right? So even if you kind of, well, we'll get there in a second. So that's a huge part of the randomness of meiosis, right? Generating that genetic diversity in the next generation. So think about this. This is a little bit mind-blowing. This is the same for both parents of, an offspring. So if you and another were to reproduce, right, and fertilize or generate a zygote, right, fertilize an egg, it would be, that gamete would be 1 in 8.4 million at least, right, not taking into account recombination. This gamete would be 1 in 8.4 million. So the number of possible outcomes from random fertilization and random alignment of chromosomes is 8.4 times 8.4 million. And the zygote is a fertilized egg. And that's diploid. Is 8.4 million times 8.4 million, which, if my memory serves me correctly, equals 70 trillion. So all of you, as individuals, you are 1 in 70 trillion, right? So if you ever want to kind of, say that you're unique, like, I'm unique, and people are like, yeah, whatever. And you say, no, actually, I'm 1 in 70 trillion. And then you can just like knock them over with science and they'll have no recourse whatsoever. Promise. Not that I've ever tried that, I've been in that situation, but it'd be fun to hear if someone has. So essentially, if you think about it that way, again, leaving out recombination, which is an added layer of genetic diversity. Essentially, you know, your parents would have to have had over 70 trillion offspring before it was somewhat likely that they'd have another one like you. Think about that. Obviously, that is never going to happen, right? It's just physically not possible. Actually, it isn't even biologically possible because you don't have that many eggs as a female. Anyway, so kind of I often use cards as an analogy. I'm not much of a card player at all. But if you think about in terms of cards, essentially, meiosis is taking two decks of cards, right? One from your mother, one from your father, or yeah, essentially, right? So these are chromosomes that you inherited when you were a zygote from fertilization of an egg, right? So the chromosomes that you got from your mother, the chromosomes you got from your father, right? Two decks of cards, essentially. And so meiosis essentially shuffles those up, right? So you might have a king of spades from your mother and a queen of spades from your father, right? It's just a giant shuffling, and then you get one of each. And it's totally random which ones you get, right? So you get the whole deck. Again, obviously, you don't want to lose any chromosomes, but it's all shuffled up, right? So it's kind of like a giant shuffling exercise in a sense. Now, recombination is essentially, if we're to take that analogy a little bit further, let's say you have a king of spades and a king of spades kind of lined up, right? So we've got one king of spades from your mother, one king of spades from your father. Recombination is essentially tearing off very precisely, but randomly, a piece of one card, tearing off the exact matching piece of another card, and then sticking them back on the other ones. So you'll have most of, you know, the king of spades from your mother, then you'll have a little bit of it that is from the chromosome of your father. And so if, you know, they're a beat up pack of cards, right? You know, you're going to have something like the scratch marks or the doodlings or whatever from one transferred to the other. So you'll end up with a chromosome that didn't exist in either of your parents. Still chromosome 1, king of spades, whatever, right? So it's still the same chromosome, but now it has a different combination of alleles than either the mother or the maternal or the paternal chromosome. Okay, so again, can't stress this enough. Meiosis is all about randomness, right? Generating the most possible genetic diversity one can, so that the next generation has a completely new combination of alleles with which to, you know, encounter the environment that it's in, essentially. So that's kind of, sorry, I got stabbed by my cat last night. My finger hurts. She's very vicious, psycho, actually. It's probably the best way of putting it. So these two big things, recombination and random alignment, happen in meiosis 1, happen in different parts of meiosis 1. So recombination, and I'll just use that rather than repeating all of them, but I mean crossing over and synapses, right? They all mean the same thing. So recombination happens in prophase 1. And essentially, it's kind of hard to present it visually, right? This image is actually, in some ways, it's a good one. In some ways, it's not ideal, right? It's hard to find exactly what you want. So recombination is a process that begins right at the beginning of prophase and has ended by the end of prophase. And essentially, you have to do three big things, right, that are specific to meiosis, right? Obviously, all the rest, centrosomes moving, spindle fibers forming, nuclear membrane disappearing, are common to all M phases, right? Mitosis, meiosis 1, meiosis 2. But in prophase, what has to happen is first, chromosomes or homologous chromosomes. That's a Scouser chromosome. Align very precisely in early to middle prophase. So if you're going to be swapping bits of chromosome, you don't want them lined up like so, right? Because then you'll end up with one really long chromosome or one really short one. That would be pretty catastrophic, right? So they have to be lined up, and they have to be lined up base by base. So ACG, ACG, right? Exactly. It's actually quite astonishing how that happens. But the thing that helps that happen is something called the synaptonemal complex. Is a big part of the alignment. Okay, so that's kind of the first requirement that's very, very unique to meiosis 1, prophase 1. Chromosomes condense as they align. Right, so again, it's hard to present this visually, right? Because it's a continual process. So they're aligning, they're condensing, and then the final part, or not the final part, that's the wrong word to use. The third thing that's happening is those that crossing over form, chiasmata form. And so these are places where non-sister chromatids, right? And they're called non-sisters because they are essentially you have sister chromatids for one chromosome, sister chromatids for the other chromosome. These ones and these ones are not sisters, right? Because they're not identical. They're different homologous chromosomes, or one or the other, right? So these are non-sister chromosomes, and you always get recombination between the non-sisters that are next to each other. These ones don't do jack. They just stay as they are. It's only the ones that are physically next to each other that recombine. And so by the end of prophase 1, you have, let's type that out, yak. You have condensed homchroms that are held together by chiasmata. Right. And so this actually is, not only does it generate new combinations of alleles, right? But also, and actually let's show you a neat kind of picture of that. Here you go. So if you have a individual that's heterozygous for 2 genes, right, on a chromosome, what a number that might be, right? So when those are copied and go through crossing over, essentially you can end up with different combinations of alleles, right? So On this chromosome, you have both dominant alleles, both recessive alleles on the other one. After recombination, now you have 4 different versions of that chromosome. You have the original, both dominant, both recessive. Those are the ones on the outside that do not undergo recombination. But the 2 non-sister chromatodes sorry, toads on the mind. I've got one in my compost heap. Chromatids, those non-sisters swap over and you end up with a new combination of alleles. So now you have a recessive and a dominant and recessive. Right, so that's a good visualization of how you get new combinations of alleles. But also, another big deal of crossing over is that I've actually typed it out already on my game, is that these chiasmata hold the homologous chromosomes together. So if you think about it, right, with those spindle fibers, they're getting pulled or trying to be pulled to different poles of the cell. But you don't want that to happen until everything's lined up on the metaphase plate. And you get the go-ahead from the anaphase promoting complex. Really, really, really important that that doesn't happen. Otherwise, you have, as we've talked about before, aneuploidy, where one gamete will have 1 too many and the other one will have 1 too few chromosomes, right? Which is generally disastrous. Not always, but in most cases. So those chiasmata, essentially you have, these 2 chromatids are stuck together. These 2 are stuck together, right? So you can't pull them apart. Now by swapping them, now you've essentially stuck one homologous chromosome to the other, right? And that's what holds them together until anaphase begins, when they get pulled apart. There you go. Sorry about that. Hope I didn't surprise anybody. Actually made my, thank you, made my eldest daughter fall over and start crying when she was a baby by sneezing too loudly, which is kind of cute. Anyway, anybody got any questions so far? Right, there's a lot to take in and I want to drink some of my coffee. So any questions, anything that doesn't quite make sense or you want explaining differently? Nope. Okay. If you do, ask. Exams on Friday. Want you to do well. So chiasmata are, good question, Infinity. And that's plural chiasma. Are the regions where part of one non-sister chromatid is stuck, onto the other non-sister chromatid. Let me see if I can, actually it's always easier to use pictures. Where are we at? Is that in linkage or is that in, I think that's in linkage. Kind of a bit of a look ahead, but oh, buggeration, that's not in this one. Not unless I'm not looking at it right. It's crazy, you would have thought I'd be. A little more. No, it's not in there either. Weird, I thought that was... Sorry, trying to think of the actual mechanism of... What bloody hell is that? All right. Sorry there, Infinity. I thought I... Can't get you over there. All right, so essentially, there you go. This is a nice looking picture. Essentially what you get is chiasmata is this region here, right? So it's the region where, and there's quite a lot more to it than that. We'll cover that later. We're not going to go into detail now, but we will later on. Essentially, this bit of DNA here kind of invades this strand, this part of the chromosome, and you end up getting a continuous piece of DNA from this black bit all the way up over here to the other side to the other non-sister chromatid, right? And you get the opposite. So there's always a reciprocal, right? So if I were able to, which I'm not, take this knuckle off my finger and stick on this one, I'd have exactly the same go from this finger onto this one, right? And so that region where those that crossing over occurs is called a chiasma, plural chiasmata, right? And so that's obviously going to get resolved, right? So it's not permanently kept that way, but at least until anaphase, essentially you have this on the left, this chromosome stuck to the one on the right by that bit of shared DNA, and vice versa. Right, because remember, here's a question. What protein holds the sister chromatids together? Nice exam question, basic recall stuff. Awesome. No autocorrect for you, Stephanie. What's the protein that destroys cohesin? This is activated by the anaphase promoting complex. Separase. Wow, you're on fire. Okay, so keep those two things in mind. They're really, really important in how this all works, right? The molecular mechanism. So essentially, we have, if I can make that, invigorate that. There you go. So we have these two sister chromatids stuck together with cohesin. We have these two sister chromatids stuck together by cohesin. So we have essentially both chromosomes of a pair stuck together because these two chromatids are stuck together, these two are stuck together, but the non-sister chromatids essentially are now shared, right, by that chiasmata, that crossing over point. So this is one continuous piece of DNA from the black bit all the way up to the gray bit, and vice versa. The gray bit all the way up to the black bit. So they're stuck together. And that's what holds the homologous chromosomes together in metaphase 1. Does that make sense? Good question, Infinity. Pardon me. Okay. Ooh, look, there's even one over here. It shows the synaptonemal complex. Super neat. Yeah. I love pictures. Pictures are great. Okay, where was I at? Okay, so you might be thinking, well, if separase destroys cohesin, right, and these homologous chromosomes are stuck together by cohesin, why don't we just pull apart the sister chromatids, right? Why do we pull apart only the homologous chromosomes to go from that diploid to haploid deal? That's the big part of meiosis 1. Right? mechanism behind that is we have cohesin all the way down, right, from top to bottom of these sister chromatids. That's what's holding them together. But the cohesin around the centromere is protected, right? It's protected by a protein called Shigoshin, which is super cool. Bang you. Bloody autocorrect. So cohesin holds the sister chromatids together and the homchroms via the crossing over. But the cohesin, is that gonna, yeah, at the centromeres is protected by Shigoshin, which is a cool word. It's Japanese for guardian spirit, which I always think is really neat. Right. And so essentially, anaphase 1 occurs and allows those homologous chromosomes to be pulled apart because only the cohesin at the tips, right, out on the arms of the chromosome is destroyed by separase. The bits holding the sister chromatids together at the centromeres is not, right? So those sister chromatids, or they're kind of sister chromatids, you know, sort of now, those are held together by that protected cohesin, which is, there's a nice picture of that. Here we go. And this is very different from mitosis. So mitosis, again, you have cohesin all the way along, but all of it is destroyed by separase. In meiosis, only the cohesin at the on the arms, right, out where you would have crossing over, is degraded, and so that allows those chromatids to stay stuck together. It's only in meiosis 2, well, anaphase 2, where Shigoshin is degraded and then that cohesin breaks down. That allows those chromatids to separate. So this is how we coordinate that, you know, that essentially diploid comes together, recombination holds them together, anaphase pulls the homologous chromosomes apart, then in this is again anaphase 1, then in anaphase 2, the remaining Shigoshin that's holding these chromatids together gets degraded. That allows those to be pulled apart. So you end up with one chromatid, now chromosome, in each of those daughter cells. It's very elegant. I like elegant biology. It's neat. It's just like, man, that's cool, right? How that happens. Okay, any questions about any of that jazz? Also, one thing to stress again is that the site of recombination, i.e., you know, where the chiasma forms, is also random. So it could be anywhere along that chromosome arm. Could be just on one chromosome arm and not on the other. Could be vice versa. Could be on both chromosome arms. It's super, super random. So every single time one of your germ cells goes through meiosis, every time, not only is it totally random which way round those chromosome pairs align? It's also totally random where that recombination occurs. Could be up here, could be down there, could be in the middle, for each and every chromosome. So if you think about it, I can't think of a more effective way in which to generate genetic diversity, short of mutation, which you generally don't want, in the next generation than what we have in meiosis. It's an astonishingly effective way of mixing up chromosomes and alleles to generate an entirely new combination. And that plus sexual, the fertilization part of sexual reproduction is what gives you essentially you, right? If you think about it, right? So this is, again, getting back to that whole why you the way you are kind of deal. Obviously, the nurture or environment part is a big part of that. But also the reason why you're not exactly like either one of your parents or a mix of the two is because of that mixing. So in each and every one of you, you have chromosomes which are just like your mother or your father, or both, and also chromosomes that are not found in either of your parents. Obviously, you all have the same, number of chromosomes. You all have a full complement, one hopes, of, 2 of 1 to 22 in the 2 sex chromosomes. But the actual exact alleles that you have on those chromosomes, some of them are going to be exactly like one parent versus or the other. Some of them are going to be a different mix, right? And that different mix is what generates those differences. Also, the fact that you're combining different alleles from your parents as well, right? So all of that is essentially what gives you I've got the famous Bartlett nose, right? Well, maybe it's not so famous. And, other particular characteristics. I don't have my dad's pointy elbows. There are all kinds of things which you can kind of see bits and pieces of how you are, even behaviors as well, right? Which have a genetic basis. Even those you see, I got a bit of that and a bit of that. But also, this thing, that's not like either of them, right? And that's because of that just absolutely complex, thorough shuffling. My wife and I do it all the time about my kids. Oh, she's just like you. No, she's not, etc., etc. Right? So essentially, that's the crux of meiosis, right? So meiosis 1 is where really almost all. I mean, there's a little bit of random alignment on metaphase 2 plate of the which way around the chromatids are. But vast majority of the randomness of meiosis is in meiosis 1. Meiosis 2, and we do go through interkinesis, which is kind of varies between organisms. So essentially, you can have either a pause where chromosomes decondense and nuclear membrane reforms and so on and so forth, like you would with mitosis. Other organisms, they just go straight through into metaphase 2, right? They kind of skip the prophase 2. It varies. But one thing to stress, though, is that all of the stuff for meiosis 1 and 2 is already prepared in interphase before meiosis, or before M phase of meiosis 1 occurs. Right, so all of the DNA that's going to be made, all of the proteins made in G1, S phase, and G2, right, before those cells commit to the M phase, right? So there's nothing made, as far as I'm aware. Well, maybe it, again, might vary. It's kind of always difficult to be like super precise about this stuff because there's so much variation. very little, if anything, is made in interkinesis between the two. And so obviously we're not done, because if you look at, there we go, right? We have two daughter cells after telophase 1 and cytokinesis 1, right? And those daughter cells are now haploid, right? That's the end of meiosis 1. You go from having 2 of everything to just 1 of everything. However, we have twice as much DNA as we should do in each of those cells. The reason being is that we have the chromatids, right? We still have, they're not identical anymore, right? Because of crossing over, but we still have our 2 chromatids. We have to split those apart. Otherwise, again, we'll end up with too many chromosomes, even though they're largely identical. So meiosis 2 is, again, it's very analogous to mitosis in a sense, because it's all about separating the chromatids. The big difference, obviously, is that mitosis, both cells are diploid, or the starting cell is diploid. Meiosis 2, the starting cell is haploid. And so really, meiosis 2 is really straightforward, right? There's nothing very kind of crazy about it. It's just all about lining those chromatids up and separating them. And so there's a couple of cool things that I want to show you here. So first up, this is actually a really good diagram in some senses, you know, kind of varies depending on what you want to show. But if you look at this, right, the centrosomes start at the top and they get pushed to essentially left and right in this image, right? And so the metaphase plate will always be exactly between them. So the plane of division is vertical, north-south, for meiosis 1. Now meiosis 2, right, we have those 2 cells, centrosomes are here. Now those centrosomes are going to move, again, to the opposite poles of the cell. Now the plane of, well, metaphase plate and the plane of division will be horizontal. So if you remember, I told you that every cell division, the plane of division flips by 90 degrees, like that. And this is a really good image of that. And so now you have your chromosomes lined up. And I always like to use, you know, fingers, right? Because they're easy to manipulate. In mitosis, chromosomes line up tip to tip. So the sister chromatids get pulled apart, or tip to tip. Yeah, like so. Meiosis 1, chromosomes line up next to each other, right? Like so, because chromosomes getting pulled apart. Meiosis 2, we go back to lining up tip to tip, because it's all about the chromatids. So you can tell, right, what stage of cell division you're at, and also whether it's, pardon me, meiosis or mitosis, just by looking at the picture. So in this picture, you know that it's, if I tell you that n equals 2, what is that, slide 10, has to be meiosis 2, right? Because you only have 2 chroms in each cell in metaphase 2. Ah, damn, my spelling is awful today. Now, if you look at, here we go, this one, oh, what's that, slide 10 of PowerPoint, what's that, 6, 5. N equals 3. Right? That has to be mitosis, because even though the chromosomes are also lined up tip to tip, you have 2 big ones, 2 middle ones, like Mama Bear, I guess, and 2 small ones. You have 6 chroms. So just by looking, and if I were to give you a question on this, I would tell you what the number of unique chromosomes are. So this is N of 3, but it's diploid, so you have 6 in total, right? You should just be able to look at this versus this and tell, okay, if this were mitosis, you would have 4 chromosomes lined up tip to tip. And it's really obvious when you're in meiosis 1 because you have chromosomes paired up next to each other. Right? That can only ever be meiosis 1. Right? So you have to be very, very precise about what stage of meiosis you're talking about. And you can tell by counting chromosomes and seeing how they're lined up in metaphase. That will tell you instantly what exactly you're looking at. Now, if slide 10, PowerPoint 5, N equals 6, right? I have 6 chromosomes. It would be metaphase 2 instead. Do you see what I mean? So just by looking at it and taking the information that I give you, you can go, ooh, that is metaphase 1, or metaphase 2. Let's bump back to the other one. Okay, and that's pretty much it from me, to be honest. There's a kind of slightly different way of looking at stuff in the PowerPoint. So I've taught you in a particular way because that's just kind of how it worked out today. It's not the only way of looking at that information. So go back through those other slides, right? Because they essentially tell you what I'm telling you, but tell you in a slightly different way. And kind of try and, like triangulate all that together to make, your complete understanding. Does anybody have any questions? Okay, so on Friday, there will not be a lecture. It's going to be the exam. The exam will open, let's type this out. No lecture on Thursday. Friday, exam will open at 9 a.m. and close at midnight. No, it's not 03, it is 04 today. Yeah. Oh, let me just finish this. Yeah, so close at midnight. Due date will be different from close date. And the big reason for that is if anyone has difficulties taking the exam, like your internet went out, power cuts, you got sick, whatever, right? You can still take it until the due date. So the due date will probably be like Sunday or Monday or something like that, right? Because the exam answers become available after the due date. So if anyone has any issues taking the exam, right, and I'm generally a pretty, easygoing person from that perspective. I know you all have different situations and the like. Hang on a sec, Stephanie. Then let me know straight away, right? Don't delay. Don't wait a week, because if you do, all the exam answers will be available to the class and you will not be able to take the exam. Let me know as soon as you can, and I will be able to open it up for you until the due date has passed. Now, don't look at the due date and go, what's he talking about? Exams open on Friday. It's actually due until Monday evening, right? That's not for you. Your exam is open on Friday, right? And Friday only. The due date is kind of beyond that, just to allow me to open up the exam if needed, right? Once that due date passes, you cannot take the exam. Okay. Good question, Stephanie. Exam will be on Blackboard. Use whatever browser you like. There'll be 1 hour, 15 minutes and 40 questions. So this exam is not closed book in any sense, right? You will be able to look up stuff on the internet if you wish. You'll be able to use your notes if you wish. You will not necessarily have time to do that, even if you could find that answer online or in your notes, right? So don't be suckered in by the fact that this is an open book, open browser exam. This is going to be time limited for most of you. Some of you might get through it quickly. Most of you will need that full hour and 15 minutes. So make sure you know this stuff, right, beforehand, because otherwise, A, you won't have time to do the more complicated questions or to look up everything on the internet or in your notes. And B, a whole bunch of this stuff isn't going to be in your notes or online anyway. It's stuff that you're going to have to work out, like chi-squared values and stuff, right? So don't rely on this being open book, open browser deal. It's not going to help you as much as you think. But do make sure you got good internet, your laptop or device is plugged in, right? Because I have had a few students go, shoot, my battery kind of kicked me out and now I'm screwed, right? I can fix that quite easily, but you don't want to rely on that, right? Just try and make sure that you have everything, kids are in one room or, relatives, you're not having to go to work like in 15 minutes, all that kind of jazz. Get it as best as you can and then sit down and take the exam anytime throughout Friday. Second thing as well is I will be around in Zoom on Friday at, 9 a.m. if you have any problems. So at least for half an hour, I don't know, it depends how interesting the news is and how many emails I have to reply to. But I'll be there for at least the first kind of half, 2/3 of class if you have any questions. And I will be around throughout the day. I can't guarantee I'll be able to answer your emails immediately, but I will be around throughout the day, barring a kind of like a 1.5 hour chunk of time after class where I'll be riding home and I won't be able to answer anything. But I will be checking my email throughout the day to make sure I can help people if they need it. I think that's pretty much it from me. Anybody else have any questions about the exam on Friday? Try and cover as much as I can so there are no surprises. Cool. All right, and if you do have any questions, and people from the other section have done this too, just email me and I'll answer them, right? It's my job. Cool. All right, study hard, which I'm sure you're doing already, Good luck in the exam, and I'll see you on the other side on Monday.